First, let me say what a pleasure it is to be back in Columbia, Missouri. I appreciate very much the invitation. As Lynn mentioned in her very kind opening comments, I was an undergraduate major in economics here, but I did have the good fortune after completing those requirements to wander over to the psych department where I met a professor who changed my life. I asked him a question and he said something I had never heard in economics, which is basically applied math. And it was that I don't know, but I know how to find out. And he taught me about experiments and random assignment and the ability to probe who we are as a species and where do we fit in the biology of life. And I've been pursuing that since that meeting here in the psychology department, McAllister Hall, as it turns out, long ago. I might also mention that McAllister Annex used to house animals, where I managed to screw up an animal study. It's probably why I turned out to study humans instead of animals. So I learned a lot at Missouri, and I owe a great debt. So it is something of a homecoming, a special pleasure to be with you today. I do want to talk about where we fit within the biology of life and what our nature is. That is the question I've been asking for almost 4 decades now. And I'll start with a description of the NSF Life Project. This is the National Science Foundation initiative. They started, about a decade ago when we knew of about 2 million species on Earth. Now, the idea of this project was to identify all the species we could before we eradicated too many more, and the estimate of the number of species around was anywhere from 5 million to 100 million. That range tells you how little we knew about the biology of life on Earth. All right? Now we know, by the way, of about 5 million species, and we're still counting. There are 2 basic design features that you find in most of those species. One, illustrated by the sea turtle down here, is that reproduction is such that you have enough offspring, some survive to reproduce. It's true of sea turtles, it's true of bacteria. If anything, it's probably the modal biological design. The other basic design feature is that you have offspring that are born ready to survive, ready to predate, ready to defend themselves early enough that they survive to reproduce. All right? Some of these animals are social, others are non-social. By definition, social animals create superorganismal structures who have function. Those superorganismal structures have evolved hand in hand with genetic, neural, cellular, and endocrine mechanisms because those structures help those organisms survive, reproduce, and thereby contribute genes to the gene pool. What I have here is a bunch of sardines being predated by a shark, as you can see. And there's a very simple rule governing the behavior of these sardines. If you look at it, the superorganismal structure is called a fishball. And these fish balls look like they have a mind of its own, right? And you ask, where is that mind of this structure? Well, it turns out it's in each of these individual fish, each of whom is trying to swim to the middle. It is dangerous for all social species to live on the social perimeter. How many of you have seen National Geographic films, right? The lions predate the antelope or water buffalo on the edge. The wolves get the bison or the elk that's on the edge. And if they're not on the edge, they move them to the edge and isolate them before they predate. Okay? It's dangerous on the perimeter. It's dangerous for fish on the perimeter as well. So the rule that governs this behavior as a structure is every fish is trying to swim to the middle. Now, there was a recent paper by a group of scientists who showed that this motivation not only allows that individual fish to be more likely to survive, but in fact, because all the fish have this tendency, they all are more likely to survive. Now, obviously, that shark's going to eat. We'll have dinner, so it's not that it guarantees survival. It just increases the likelihood as a set that they survive. This led to a very simple principle. It was unfortunately espoused in a book with the name The Selfish Gene, which brought with it all kinds of implications for how organisms and humans think rather than how genes operate. It could have been called the Xerox machine, and it would have had the same meaning, but the impact, the misunderstanding by the public wouldn't have been as great. Here's the basic premise. Whatever the contributions made by a gene or set of genes to an organism's structure and function are passed on to the next generation, if and only if the gene makes its way to the gene pool. Those sardines who did not have the motivation to swim to the middle and were on the edge were more likely to be predated. Guess what? That functional tendency disappeared as they disappeared and their offspring never occurred. All right? So you have here a notion of survival of the fittest. But that, when we talk about survival of the fittest, we're not talking about genes, we're talking about organisms. And I want to emphasize that this is true for the genes, not for the organisms. All right? Now, for years and years, we studied individuals in isolation. We did that with very good reason. We did that because we wanted to control all extraneous variables. And if I have a group in the lab, God knows what they're doing to one another, right? So I want to isolate them and study them, even as a social, if I'm interested in social questions, so I know and can control everything to which that subject is being exposed. So whether I'm studying animals or humans, that kind of isolation has been important. I think with that, it was easy to forget the importance of these connections to others in our species. And so how does one examine this? How does one examine the impact? In particular, I was interested, as I mentioned, in the neurocellular, hormonal, and genetic mechanisms underlying these structures as well. That's basically what social neuroscience is about. Social neuroscience, when I first proposed it, I put a colon after it, and I put the phrase an oxymoron question mark. Because one could reasonably ask, and neuroscientists were certainly inclined to think of it as something that made no sense, since you're asking about processes that extend outside an organism's skin. And usually, if you go outside the membranes of a cell, you're getting a little loose in neuroscience, all right? So I thought the best way to do it was, in fact, to take a traditional neuroscience approach. If I want to understand what a gene is doing, I look at a set of mice with that gene, a normal set of mice, and a knockout mouse group. And that knockout mouse is identical to the other, except for that one gene. And I look in differences in function or structure or operation as a function of that gene deletion. I'm not interested in the knockout mouse per se. I actually am interested in the gene that's not present in that knockout mouse. But I study it by looking at its absence. What is the effect of its absence? Or if I want to know what the orbital frontal cortex does, I might look at Phineas Gage's behavior before and after a tamping iron obliterated his orbital frontal cortex. And from the changes in behavior, or in lesion studies that are now done, temporary or permanent lesions, you can find what functions are associated with that because of the absence, because of the changes in behavior associated with that loss. So what we did was to simply look at individuals with and without those connections, thus the total social isolation. It's not that the target of interest is social isolation. It's really what is the function of the connections that define who we are as a species, as a social species. To study their function, think about isolation as like studying Phineas Gage. I'm not actually interested in a hole in the brain. I'm interested in what fell into that hole, what was obliterated by that tamping iron. So I'm interested in the connections. The way to look at what are the neural, hormonal, cellular, and genetic effects of these connections is to look at their absence, contrasting it with their presence. And so that's the approach that we took. And that's the approach I'd like to tell you about. Now, this is a graph from House, Linus, and Umberson's 1988 science paper. They had already documented that social isolation, defined as a lack of being married, a lack of frequent contact with friends and family, a lack of voluntary association participation, and a lack of belonging in churches. That's socially isolated in this context. Doesn't mean they're alone. It means they simply don't have those types of connections. Is associated with broad-based morbidity and mortality across different prospective studies. Science 1988 paper. They argued that the reason for this association was because of something we all know affects your health and outcome, and that is your health behaviors. If you have friends and family, spouses and churches encouraging you to take care of yourself, you engage in better health behaviors. If you engage in better health behaviors, you're likely to survive longer, suffer less morbidity and mortality. All right? Well, I read this paper and I thought that couldn't possibly be the whole story, if any of the story. And there's a couple reasons for that. One is if you look at non-human animal research, where you randomly assign genetically identical animals, going from primates down to invertebrates, what you find is those social animals, when isolated, die earlier. In a paper that appeared in Science Today, McIndoe and his colleagues showed that in the mouse, you isolate that developing mouse for 2 weeks during a critical period, and the myelination of the prefrontal cortex is compromised, the molecular mechanisms are specified, and that deficit is never recovered. As a function of that, that mouse's cognitive capacities are limited for its whole lifespan. For the fruit fly, you isolate a fruit fly, it dies earlier, it's due to oxidative stress. We've recently done a review of all of this literature. The stunning thing to me is how similar the mechanisms that kill these animals are in what we see in humans, as I'll show you. For instance, in humans, we see differences in the gene transcription expression underlying oxidative stress. just like you see manifest as a cause of death in these fruit flies. There is something fundamental, something very rudimentary about being a social species and finding yourself in the absence of these connections. Now, when we're dealing with non-human animals, you can randomly assign them to be in a cage by themselves or be in a cage with another animal. This study with mice, it was an artificial stroke, cerebral ischemic event, identical in all animals. Some of these animals are pair-housed, others are randomly assigned to be individually housed. The stroke is effectively the same. The neural damage is 3 times higher in the isolated mouse. All right? We know that loneliness, isolation predicts over time the onset of dementia and cognitive decline in older adults. It's not plaques and tangles. It's probably this. Oh, I should mention, you block the inflammatory response in this animal, interleukin-6, and you eliminate the difference between the isolated and the group house mouse. In humans, it's not just interleukin-6, it's a whole family of pro-inflammatory cytokines or markers associated with this, and it can be associated with neural damage. And so you see, we get an elaboration, but the very systems are constant across these. So as a preface, what this suggests is that there will be brain mechanisms that are triggered that involve, and not at all, your awareness or your consciousness, all right? So just as a premise, you might expect that from this. The other thing is, it can't be social control. I don't think the fruit fly is dying earlier because its spouse or friends are telling it, you should get some exercise. It's not the health behaviors, right? Social control doesn't explain these animals' earlier death, all right? Okay, a second is because this emphasized objective isolation. Yes, we too see something when we look at objective isolation, but being psychologists, we also asked about perceived isolation. We can't take humans and randomly assign them to a closet for 10 years or for 3 weeks, right? That's just not allowed. And so we've looked at whether you feel isolated, and indeed in humans, being alone or being with others isn't really the important critical feature. Freshmen who come into the University of Missouri, that first week or two, they're actually around more people, perhaps sharing a room for the first time in their life in larger classes than when they were at home, but they feel more isolated. It's whether, in fact, it's miles from home that we found to predict how lonely those incoming freshmen feel, all right? So this perceived isolation is more important than the objective isolation when you allow your participants to select their own way into situations, all right? It's not like the animal studies. If indeed we were able to randomly assign people into isolation conditions, that's what we see in the animals. But because we don't, it's whether they choose to be alone or not. That's what's critical, all right? And so we look at objective isolation, we look at perceived isolation, and again, we see perceived isolation to be the determining factor. And these are just examples. This small child might have someone with her, but the fact that a parent just left is going to make her feel isolated, whether or not they have loving family around. This kid is around other students on the school grounds, but can feel, if you were ever the last one selected in a sports team, like this kid might be, or left out, you know that the feeling of isolation can be intense, in fact, exacerbated by the presence of everybody else. And this is Pat Riley, as many of you know, the general manager for the Miami Heat. As some of you know, the Heat and the Oklahoma Thunder were in the NBA Finals. I thought this is from game 1. The Oklahoma Thunder owner has a sense of humor. They placed Pat Riley in the middle of the Oklahoma Thunder fans. This is when the Thunder were winning this first game. And Riley is with a lot of other people, but I would suggest he's not exactly feeling connected to anyone around him. All right? So let's just emphasize that it's this perceived isolation that's really the critical factor. Oh, I'm sorry, I meant to show you this slide. I found myself studying loneliness. Perceived isolation had already been defined as loneliness. All right? So I didn't set out to study loneliness. I had this other question. And because there was already a massive psychological literature on this topic, we simply adopted the term loneliness rather than pretending we were studying something new. But I'll tell you, at the time I started, loneliness was thought to be, as he said, chronic disease without redeeming features, a little different than depression or introversion or shyness or poor social skills. All of those turn out to be untrue when you look at population-based samples. All right? And we've been working with population-based samples, by which I mean the same kind of sample the news media draws when they're trying to predict a presidential outcome. It's a random sample of humans in the United States so they can generalize to everybody else. We don't take extreme samples in the studies. Most of the studies about which I will be speaking, we take these population-based because I want to say something about we as a species rather than about people who are very lonely. All right? So we get this whole range, and that's the primary population I'll be talking about. This is to show you that objective isolation as measured by sociologists do in fact map into whether you feel isolated or not, but it's a weak association. It's far from a linear relationship. Okay, this is simply to show you that felt isolation, loneliness, predicts who dies. All right, net a number of other factors. So in this first model, what I'm showing you is that how lonely individuals in the health and retirement study, a nationally representative sample of older adults in America, it predicts higher incidence of death in those individuals. All right, now that's over a 6-year period. And you see, we've got other factors we're also controlling. So men are more likely to die than women. The older you are, the more likely you are to die. Ethnicity, not significant. The richer you are, the less likely you are to die. These demographic factors are known to be strong predictors of mortality. So this effect is above and beyond, for instance, being old and being a male who's poor. In this second model, we've added the objective social circumstances. And you notice that loneliness continues to predict who's dying above and beyond the objective social circumstance, what I mentioned just a moment ago. What you see in this third model is now I've added health behaviors. And you notice these asterisks, health behaviors do predict who dies. So they are very powerful predictors. When we talk in health psychology about trying to change somebody's health behavior to be more positive, yeah, it has positive health consequences. That's not a shock. But look at what happened to the ability of loneliness to predict mortality. It didn't change at all. When you add predictors, even error variance is going to increase the variance you account for. This is a nominal reduction. There's no real change. When we control for other factors like functional limitations, self-rated health, and depressive symptomatology, outcomes we know loneliness affects, so I'm overcorrecting, you see that even then we get only a nominal drop in this ability to predict mortality. All right? So this is a powerful variable in terms of predicting above and beyond these standard health factors. This is another, we published the first study just this year, late last spring. This is one that came out in the summer that basically shows some of the same things. Death, you notice, this is all loneliness predicting this. And these are the morbidity statistics, ability to climb, upper extremity tasks, mobility, ADL. So you see that loneliness is also predicting those who are becoming less able. So both morbidity and mortality. That's why I mentioned when we control for these factors, we know loneliness to be producing the outcomes. We can control for these factors and yet find loneliness above and beyond these, still is predicting who is it that's dying. Okay, so what is loneliness? Well, there's a couple different ways to answer that question. One is what's the phenomenology of loneliness? That's what you experience, not all that useful. What we've done is to look at what is the, if you will, the mental representation, not your conscious representation. What is the mental structure? If I asked you, what's the structure of rooms, you might tell me and we'll get a lot of variance. If instead I ask you a lot of questions, and then I put those questions into multidimensional scaling or factor analysis, what I'm likely to find common to all of your descriptions is rooms have width, depth, height, and stuff, right? That would be what was common across all your descriptions. Well, essentially, think about talking about not rooms, but your connections with other people. We subject that to both factor analysis and multidimensional scaling, and we find 3 dimensions. One of those dimensions we call intimate connection or intimate isolation. In the traditional psychological literature, this has been called emotional loneliness. These are the kind of items that tap where you fall on this continuum. Gary Larson cartoons, he always has a way of capturing succinctly what it is that scientists study. In this particular one, you see these 2 individuals who aren't necessarily closely linked. You say, hey, come on now, you 2 were made for each other. It is the sense in which somebody else affirms you have value, you have worth, you have meaning. All right? People who are elderly and isolated, one of their biggest existential concerns is that they will die and no one will even know it. They would be very low in intimate connection. All right? Now, we looked at what is the best predictor of this dimension, net the other 2 dimensions that I haven't told you about yet, and it's marital status. Marriage is associated with lower levels of loneliness, in particular this dimension. But marriage doesn't guarantee intimate connection. So this is an example of a married couple who's low on that. And in fact, A colleague of mine at Chicago, Linda Waite, has found marriage is health protective. Well, when we and she looked at it more carefully, again, looking at the health and retirement study and our own data from the Chicago Health, Aging, Social Relations, another population-based longitudinal study, we find marriage is health protective for 2 reasons. 1, it makes you less lonely. It gives you this connection. Number 2, on average, not everybody, but on average. Number 2, household income is higher. And those each have independent health protective effects, all right? If you are in a marriage and you're poor and you don't feel close to your spouse, you have no health benefits. In fact, there was some suggestive evidence that it might be worse for you because essentially you're in a bad circumstance and it's harder to correct than if you have no relationship. Okay, a second dimension is what we call relational connectedness in the psychological literature. This has also been called social loneliness. It's what most lay people think when you mention the word lonely. It's, well, what's your contact with other people? Again, this cartoon kind of captures it. You have these 2 people here. You can imagine it as graduate students who are sharing an office or faculty who have adjacent offices. Thanks for being my friend, Wayne. It's not guaranteed you're going to like that person, right? If you do, the world is a lot better place. These are the kind of items that we use to tap it. Now, you will notice in no item will you see the question lonely. We don't ask them, do you feel lonely? If I ask, do you feel lonely? The males in this room would go, nope, nope, right? Because it's like wearing a big L on your head. I actually, from the literature, it's clear that loneliness is stigmatized, particularly in males. I didn't actually feel that. I knew it cognitively until I wrote a book. And across the front was loneliness. I'm on a book tour. I'm on a plane. I placed that down on the plane's table. And other people around are looking at it. And then I understood what that literature said. Then I felt. I knew it cognitively. May I tell you, the emotional understanding is profoundly different. I quickly turned the book over. I'm the author. I'm the one. All right? So that's what people have to deal with. For that reason, we don't ask people, do they feel lonely? We ask these kinds of questions. We know them to tap it. People try to connect with other things besides people when they have no relationships. We've even studied pets, so I can answer if someone wants to know. I can tell you the answer to that question as well. The best predictor is the frequency of contact with friends and family. That's the best predictor of where you fall on this relational connectedness. The third facet hadn't been identified in the prior work on loneliness and psychology, and we call it collective connectedness. Remember right after 9/11 that there was a sense of camaraderie in America. There was a sense of treating each other nicely, so much so that it made the national media like, what's going on? Well, that identity as an American was made salient by that attack. And it is that identity that then started to have effects on everybody's behavior. Now, of course, admonitions from the White House that we should watch for evildoers in our backyards kind of helps to undermine that trust we had in one another. But that's effectively what collective connectedness is. It's this feeling that you have a broader identity with others who may or may not be personally known to you. All right? I live near the Chicago Cubs Stadium, Wrigley Field. If the Chicago Cubs, if they just do well, it becomes insane. All people who have Cubs insignia start to become friends with one another. If you watch the Olympics this summer, you probably saw the news that people in London at the pubs were getting up and high-fiving each other and were friends. They didn't know each other except they were all rooting for the London athletes. All right? That's the power of this collective connection. I think Gary Larson really nailed it. In this particular cartoon, you have these 2 bears, perhaps on Wall Street, and they're obviously friends, right? One turns to the other and says, well, we're lost, and it's probably just a matter of time before someone decides to shoot us. All right? That's the absence of collective connectedness, even in the presence of, if you will, relational connectedness. The best predictor of that we found in our longitudinal population-based samples is number of group memberships. I have to tell you, this kind of link between antecedents that it's supposed to theoretically be related to and what empiricism actually show seldom occurs in my laboratory, so this is something of a surprise to us. Now, we've looked at this a little more carefully and asked, how generalizable is it? And I can tell you, we find the same structure in men, in women, in young adults, in old adults, in African Americans, Euro Americans, Latino Americans, and in Beijing in both young adults and in older adults. It's the same structure. So I don't know if it's universal, but it's profoundly generalizable so far. All right? So I have this simple model I'm going to articulate, and then we'll move on. Here's the first premise, one you've already seen. Genes that promote behavior which increases the odds of the genes surviving are perpetuated. Notice we're vehicles for genetic transport. So it's not our behavior, it's the genes that are critical here. When offspring have long periods of abject dependency, selfish genes incline social brains and bonds. Your genetic legacy does not, in fact, this is true of all mammals. We have the longest period of abject dependency of any animal. It is not your ability to reproduce that determines whether your genes make it into the gene pool. Now, most assuredly for bacteria and sea turtles, that's probably the case, but not for humans. If you are in conditions of privation and you're very selfish, you're Homo economicus, right? You're very selfish and only concerned about your own behavior and your own survival and your own hedonism. And you have a child who needs protection and needs food, but there isn't much food. So you don't engage in any self-protective or bonded behavior. You only engage in your own self-preservation. You're likely to survive and have more offspring, but your genes will not make it into the gene pool because none of your offspring live to have offspring of their own. In mammals, it's the ability to become a grandparent because the infants are dependent on you for survival. All right? And so that's the reason for the genetic transportation that's important. Right? It isn't your ability to reproduce, it's whether those genes make it into the gene pool. And those infants can't reproduce yet. And if they all perish, then your genes don't reproduce, all right? So you can see how one trial, one generation of this, and you wipe out that genetic tendency, all right? The pain and dysphoria of social isolation evolved as an aversive biological signal to promote opportunities for the social connection necessary for one's long-term survival and for the survival of your genes. Okay, so it's just like pain. Physical pain protects your physical body. I don't want any of you to be in chronic physical pain. I also would not wish on you that you had no pain system. Because people who are born without a pain system, it's very difficult to keep them alive and from having a very difficult life because they don't know when they're doing something that's damaging themselves. When they're breaking something, when they're cutting something. And so it's really, really difficult. That pain system protects your physical body. Loneliness protects the social body we also need as a social species to survive and prosper, right? It works identically. The aversiveness causes you to stop doing what you are doing and to do something that's more beneficial for that social body. In theory, that was true across evolutionary time when we lived in small, static villages. We live in a very different society today. It also, however, motivates vigilance for assaults that threatens one's short-term survival. We see this in non-human as well as human animals. And this sounds like it's in contradiction to the first, and in part it is. Being on the social perimeter is not only sad, it's dangerous. And so your brain, whether you know it or not, your brain goes on alert for social threats. Remember Caesar's last words, E tu Brutus, right? He misjudged the social context. He didn't feel isolated. If he had felt isolated, he might have been a little more cautious around Brutus as well and might have lived to survive, all right? So in fact, if you take a rat, you individually house it, you put it in an open field, rather than walking around the whole field, as is typically the case, it walks around the outside walls. It's called predator evasion. Well, I'll show you that humans' brains are basically doing the same thing. And then finally, it actually facilitates perspective taking and social learning in ways that I'll try to just mention at least. Okay, how many saw March of the Emperor Penguins? Okay, so I'll keep it very brief. These are strange animals. They live by the ocean where their food source is, but when it comes time to hatch their chicks, they march 70 miles into the middle of the Antarctic because there's no predator dumb enough to follow them there, where it's minus 70 degrees and over 100-mile-per-hour winds. The mothers hatch the chicks. They're placed under the abdominal fat of the fathers, and the fathers sit there throughout that winter or summer until the chicks are large enough and they all march back to the ocean where they spend most of the year again, all right? These structures, though, that they create are called huddles, and they're necessary for genetic survival. So the mother was necessary, but the mother went back to the ocean. She didn't stick around. There's no mothers there. These are only fathers. Because they were depleted, they needed to go eat. The fathers, primarily, you notice, if you can see this, they all have their back to the outside because it's viciously cold. And they're primarily sleeping because they try to preserve their energy, all right? But they also have, if you've watched the film, it looks like they take turns, they rotate in. Biologists, since the filming of that, have documented what they call cooperation among these penguins, and that it's not like the fish who are trying to get to the middle, they voluntarily are rotating in. Everybody's taking a turn on the outside because that allows all of them to survive. If you're isolated and only on the outside, you'll perish. There are occasionally penguins that get ostracized, they perish. All right, they don't make it. Their chick and the father both die on the ice. All right? So that behavior promotes all of their survival. So the mother's necessary but not sufficient. The father's necessary but not sufficient. The huddle is also a vehicle for genetic survival and transmission. All right? Now, if we're going to study this in humans, one of the things that I was concerned about early was to what extent am I looking at just weird or low social capital individuals? This is a picture of Russell Weston. Russell Weston was the individual about a decade or so ago who walked into Congress and started shooting people. And like Ted Kaczynski, he was something of a loner. And so there's this notion of people who are alone, kind of odd people, that's really what we're dealing with. And indeed, if you look at lonely versus non-lonely undergraduates, this is from the top and bottom quintile of incoming freshmen at Ohio State. There's about 2,500 students in this study. This is just the top and bottom quintile. Obviously, they've been selected to differ on loneliness. They also differ on social support. So lonely individuals have less social support. They're shyer. They have poorer social skills. They're more hostile. They're more anxious. They have lower self-esteem. They fear negative evaluation. They're pessimistic, and they're in a more negative mood overall. Which of these is the bus driver and which of these are passengers? We worried about that, all right? And so there are a couple ways we've been trying to address these other variables that are co-varying with loneliness. One, we measure all of these and a lot of other variables, and we statistically enter them into the equations before we enter loneliness. So we've corrected for these influences, all right? When we can, we do experiments. So occasionally I'll show you where we randomly assign people to lonely or non-lonely conditions. We have a couple of nefarious ways in which we've manipulated loneliness. I'll show you one in just a moment. We do that when we're looking at short-term effects. If I want to know what is the mechanism by which loneliness is leading to early morbidity or mortality, we obviously can't manipulate and keep you lonely. All right? So for that reason, we do longitudinal studies with all of these. So I'll be showing you cross-lag structural equation models in a longitudinal analysis where we're controlling for alternative interpretations and then looking at whether loneliness, above and beyond all these other potential operating variables, are contributing. And I mean by that this set of variables as a case. All right? And finally, animal models. And we're increasingly doing some animal work, and we now actually have a non-human primate model, not only of isolation, but of felt isolation. And I'd be happy to answer questions about that during the Q&A. It's been kind of an interesting project. But let me show you an experiment. At the time I did this study, the MacArthur Network that helped fund this original project was reigning, and this is Steve Coslin. Steve was in the network, so was Dave Spiegel out at Stanford. And Steve and David just published a study on experimental hypnosis, showing that high hypnotizable subjects who saw either this kind of picture or an identical picture in monochromatic presentation told that it was color or black and white. The high hypnotizable subjects, the area of their brain involved, they were using PET, involved in color perception, was active based on what they were told. Low hypnotizable, it was based on what they saw. Now that was the best study, the most compelling study of hypnosis I've ever seen, all right? So we flew out to Stanford, which is where this study was done, and David Spiegel did the experimental hypnosis of those 20 high hypnotizable subjects in this study. So I didn't want to let anything go to chance, right? They demonstrated that these 20 subjects, high hypnotizable subjects, that the brain was active not based on what they were actually being shown, but what they were told were being shown under hypnosis. So we went out and used them, and we manipulated in a counterbalanced fashion across subjects whether they felt lonely or non-lonely. And then we gave them these scales, all right? So the reason I put this on one plot is I want to show, all of these are significantly different, but I want to show you what it looked like when we experimentally manipulated it within subjects, right? Whether we made them feel lonely or non-lonely first didn't matter. So I've collapsed across that order, that blocking variable. Oh, I have it this way. Oh, yes, I wanted to point this out. I showed you the variables that our undergraduates differed on. We thought they would also differ in terms of their impact to emotional events. And this was Horowitz's score. And what we found was they didn't differ on that. Our undergraduates who were high and low in loneliness didn't differ in this particular response to stressors. All right? Neither did our subjects at Stanford when we experimentally manipulated. I wanted to show you that because it's not just that they gave us all possible differences. If they're role-playing, you'd expect them to also differ. This looked just like our undergraduates. This was the rest of the result. And what stunned me was not only that we got identical differences, but the scale values themselves were stunningly similar. When you feel lonely, you feel shyer. You feel like you have poor social skills. And I'll explain why that is as soon as I show you a brain imaging study. You feel more negative. Your self-esteem plummets. You feel more depressed. We found this factor to drive a whole set of other factors, much more than we ever anticipated. All right? So this is the kind of thing we do in experiments, and it also suggests a centrality to this variable that we had not anticipated. So I showed you one kind of overarching model. Let me show you a little more detail. Perceived social isolation leads to a motivation to connect and an implicit hypervigilance for social threats. Implicit because we see it, first of all, you're not aware of it, but secondly, we see this same process in low-level animals that we're not thinking or engaged in self-reflective thinking. That affects attentional, confirmatory, and memory biases. If my brain's on alert for a social threat and I'm interacting with you, I'm more likely to see it in an ambiguous behavior. That's going to lead to behavioral confirmation processes producing more negative displays and expressions. You can see how that's going to go play in the town, right? And then, of course, that can reinforce perceived isolation. Even though isolation is designed to promote connection, it does so in this kind of cautious, guarded way. If I don't get out of it, that then activates neurobiological mechanisms that can lead to early morbidity and mortality. That's the overarching model. Let's look at some pieces of it. Well, one of the things that this, you might say, this sounds very deleterious. Why did this evolve? Right? And one of the things is it keeps you in a safe circumstance even when you're on the perimeter. That is, you don't create Caesar's error. But it also facilitates social learning. Now, what I'm showing you is a child in timeout. I'm not suggesting that timeout is a practice that has evolutionary significance. What I am suggesting is that shunning and ostracism have been practiced for millennia across cultures, right? Why? Well, when do we put children in timeout? Just as a model for the answer to that question. It's when they're acting narcissistically, when they're acting selfish in a social context, right? Your child is with other children, dominating all the toys, they're not playing well, so we say, okay, no more of this, 1 minute per your life, put you in a chair by yourself, right? And I don't know about you, but when I did that with my child, I'm going 1 minute of your life, I can do this. This is hard. Because what do they show immediately? They show expressive displays or acoustic signals that they're sad. And those are safe signals. They basically serve as a call for reconnection. And I don't know about you, but I, as a parent, felt that call very strongly. And it was me who had to engage in impulse control, right? You want to go grab the child and pick them up and comfort them. Go, no, 1 minute per year of life, I can do this, right? So that negative display is, if you will, an evolutionary adaptation that is a safe call for social reconnection. If that child instead were to not be somewhat depressed and force their way back in, then damage done to the individual, all right? So you can see how this tendency of depressive display promotes that reconnection when possible. And what you're doing, you take that child, you hug them after the 5 minutes, let's say, you put them back with the other children, what do? They play better, right? Notice you just saw an example of social learning being facilitated by the loneliness that they felt when you rejected them, all right? That's an extraordinary, social learning is perhaps the most important difference between humans and other species. We do social learning better. Yes, we have opposable thumbs, so do some other animals. Yes, we have an upright posture, not really going to explain everything. Social learning seems to be critically important, and this allows you to train your offspring to match whatever culture or social context they happen to be born into. A very powerful adaptive tool, all right? So that's one of the ways in which loneliness has a very productive effect. Now, I've already suggested that the experimental manipulation makes you feel depressed. Well, loneliness and depression covary, not a surprise. What's causing what? We asked that in our Chicago Health, Aging, Social Relations. This is a population-based sample of individuals born between 1930 and 1952. I started the sample in 2001. We're now in year 10. We skipped 1 year. We're in year 10 of data collection from these individuals. This is just the first 5 years. What I have here is loneliness, predicting subsequent years of loneliness. The prediction here is about 0.86. The test-retest across a year is about 0.77. These are non-standardized coefficients. Depression is the best predictor of how depressed you are next year. That's not a surprise. Those are the first things we enter. We then enter a number of demographic factors and standard medical risk factors, psychoactive drug use, treatment, and here I also have entered perceived stress. And what you see is loneliness is predicting increases in depressive symptomatology above and beyond everything else, and depressive symptomatology is not predicting increases in loneliness. If I do not control for these other psychological processes, it's reciprocal. It looks reciprocal. When I add in other psychosocial factors that I showed you, then it's unidirectional, and it's loneliness that's leading to increases in depressive symptomatology. Right? Well, I suggested that loneliness is associated with this hypervigilance for social threats, implicit no less. That's a pretty bold statement. One of the ways we tested it was the Stroop test, and you know this test. What I'd like for you to do is to look at the letter strings I'm going to present and tell me what color each letter string is in. Yes, the letter strings are words. Don't read the words, that will only interfere with your performance. Just say what color they're in, all right? Blue, orange, green, red. Easy, right? Okay, let's do it again. Exactly the same task. Ready? Harder. You feel it's harder. Why? That's because your brain is reading the words even though you don't know it. You're trying not to. Your brain's doing it despite what you consciously think you're doing. And that's what I'm arguing the brain is doing when you feel isolated, is it starts surveilling for social threats. You have no control over this. So how do we test it? Here's what we did. We presented words like this: negative, non-social words, and negative social words. Because negative words tend to take longer to name the color than positive because they attract attention. So I'm going to control for how negative these words are. Social, non-social, I'm going to subtract and see what's unique about negative social, right? And positive social and non-social words. Is it true of surveillance of any social stimulus? Maybe loneliness makes you attuned to all social stimuli. Or is it true just negative social? Here are the data. This was from letting loneliness very naturally. The lonelier were the individuals, the longer it took to name the color of the negative social words. Now, you can't tell, it's just this is negative social versus non-social, but in fact, it's the negative social that's taking longer. We've also done experiments where we randomly assign people to conditions, we make them feel lonely or not, and you get the same effect. So this isn't some other trait that's operating, it's having put the brain on a loneliness alert that induces this hypervigilance. It's what Caesar didn't do. We've also done fMRI work on this, exactly the same idea, except instead of words, I'm using pictures. So negative non-social and negative social pictures, positive non-social, positive social pictures. Simply do a contrast of this or this, and we convolve that, if you will, hemodynamic response across the brain with the loneliness felt by that participant. Basically, the question is, how does the brain of a lonely versus non-lonely person differ in their response to negative social versus non-social pictures? Okay, if you can wrap your hand around that, that's what we're asking. So we'll look at the negative first. What we see are two primary areas. The yellow area is your visual cortex. When someone who feels lonely is looking at this versus this, visual cortical activity is greater when looking at the negative social picture. This is essentially a conceptual replication of the Stroop test I showed you. Negative social, it attracts more attention in the brain of that individual. Now you follow that visual stream forward and you get to this kind of strange area called temporoparietal junction. I say strange because as a junction, it's actually an area between brain tissue instead of brain tissue itself. But that surrounding area of tissue has been associated with perspective taking, theory of mind, empathy, and essentially you can think about it as an area of brain tissue that's really responsible for an incredible magic trick. Attentional control that allows you to take your focus of attention from inside your skull and simulate being inside someone else's skull to take their point of view. All right? That's lower when you feel lonely. Why? It's dangerous on the social perimeter. These brains are concerned about self-preservation. If there's a negative social event, your brain goes on alert. Now it's concerned about protection of you. Not that person's dire condition, but the damage that may occur to you. And so you get less of that. When we look at social skills, lonely individuals have the same social skills as the non-lonely. We're looking longitudinally, it's the same person. They didn't lose social skills. But if I'm thinking about myself rather than you, I'm not going to exercise as appropriate to social skills. So you express poor social skills. It's because you become more concerned about self. The brain, well, becomes more concerned about self-preservation than the concern about others around you. All right? Now, this is just to show you, if you look at this area, it nicely maps into areas that have been identified with theory of mind and in perspectives taking. What I wanted to show you down here is we did experience sampling methods, and what we find is that lonely individuals and non-lonely individuals, at least young adults, are in very similar circumstances. But the hassles that are incurred are felt to be more extreme by lonely than non-lonely individuals. Okay, let's do the same for the positive pictures. You see a very different area of activation in the brain. This is a sizable area. In fact, this is the area that you get active, which is right about here when you engage in cooperation. This is a study of love. It's about here. This is the dopamine-rich area. And so we get ventral striatal dopamine area really lit up when you look at people versus equally positive, visually complex and arousing pictures that are non-social. And the effect is such that the less lonely you feel, the more ventral striatal activation you're seeing. Let me put it in a more apocryphal fashion. You're sitting at dinner at a restaurant with someone you absolutely adore. You are joyous together. You see a lovely, loving couple walk in the door. You say, that's a positive picture, or scene. That's a positive couple. And you feel the joy that they express. You are at that same setting, and you're with that same person, but you feel alienated and distant from that person, right? I know you've never had this experience, but you might just nevertheless try to role play that, and you see that same loving couple walk in. Well, you can still say, I know that to be a positive picture. That's a positive scene. They're obviously happy, but you don't feel the joy. That's basically what this brain response is showing. As a lonely individual, they actually show greater ventral striatal activity to the non-social pictures. When you actually break this down by cell, and the lonely are feeling greater here, the non-lonely greater activation to that, even though both groups rate both pictures equally pleasant. All right? Now, overall, they're showing activation of the ventral striatum to both sets of pictures. So we're talking about the differences in activation, the differences in pictures. And what I didn't show you was, fortunately, even for non-lonely, most of our encounters are positive, but the lonely, they rate those positive encounters as less uplifting than the non-lonely. So it's basically what you would expect by this result. Okay. One of the implications, and I alluded to this earlier, is that the conscious experience of loneliness can have a variety of effects. Don't get me wrong. But the brain has been sculpted to respond to perceived isolation, even in the absence of consciousness of being lonely. Right? And I thought, well, the studies I've showed you make that argument, but how could we try to demonstrate this beyond what I've shown you? And I thought, sleep. When you're asleep, you're not sitting there deliberating about anything. There's no reflected cognition in my dreams, at least, right? So what do we see during sleep? Do we see lonely days invading the nights? So we took our undergraduates. We were bringing them into the hospital, and these poor kids went through a lot. But we also had them in the hospital overnight. So we measured their sleep. The nurses didn't know, the circumstances were identical. The nurses didn't know who was lonely, who non-lonely. In fact, the kids running the study in the hospital didn't know who was the lonely or non-lonely participant. All right? What do we find? Oh, I might mention, we find differences in sleep as well in other animals. But the way to put this framed a little bit differently is, if it's dangerous to fend off wild beasts with a stick by yourself, imagine how dangerous it is to lay that stick down to go to sleep when predators are out and you don't have a safe social surround. Does the brain remain on alert for threats? And so we tested that. And what we find is that for those lonely undergraduates, they showed the same amount of sleep, but they had more micro-awakenings. Sleep efficiency was lower, all right? Micro-awakenings are these you kind of startled and you go back to sleep, you don't even know you had them. However, you wake up in the morning more fatigued. If you've ever gone to, say, to Chicago or New York, and you stayed downtown, you probably showed a lot of micro-awakenings because the context is different, it's noisier than here in Columbia, and you wake up the next morning, you're tired. Now, remember, the day after that, you're sleeping through it. But that first night effect, you can have these micro-awakenings. It makes you fatigued, all right? So we measured not only in the hospital, we also measured 5 days in their dorm room to see if it was just a first night effect. We got the same results. Also, in that undergraduate sample, use what's called the Pittsburgh Sleep Quality Index. And what you see in our undergraduates is they're complaining about being tired during the day. They're complaining about having poor quality of sleep, taking longer to fall asleep, not sleeping as long. In fact, they are. They just feel tired in the morning, so they're assuming all of this is true. All right? The reason we used this was because as we went to our older adults, you can't do the same things to the older adults that you can do to your undergraduates, especially if you want them to ever come back in a longitudinal study. So rather than hook up all the equipment to measure their sleep, we gave them the sleep quality index measure. And what you see is an exaggeration of sleep disturbance. Not only are there even larger differences, but they're also taking more sleep meds as older adults. But it's not helping. So if there's a deleterious effect from your brain feeling threatened during the day, you're also not detoxifying those days, stressful days, as much across a lifetime. And that also can contribute to morbidity and mortality. All right? Now, I mentioned we also have done longitudinal research. This is a case where we're looking across 3 consecutive days in our Chicago study, and how isolated they feel that night is predicting the daytime dysfunction they feel the next day. But the daytime dysfunctioning isn't predicting how isolated they feel. All right? So, and that's controlling. This is just a partial list of the covariates. It's controlling for social support, depressive symptomatology, since depression is supposed to be a predictor of poor sleep. We're controlling for that, and you see this above and beyond. Now, I'll show you one other study, because all of these studies have involved urban samples, and I have given you an evolutionary story. Well, it's hard to find conditions of privation, but we have found are very isolated religious cults. This is the Hutterite population. It's in western US. They're among the most communal. The level of loneliness in this group was the lowest we've seen anywhere in the world. And we've studied Europe, Asia, and North America. And their overall level, they're highly communal, they're highly insulated, they're highly agrarian. Nevertheless, there is some variance in loneliness, but it's minimal compared to what we've seen elsewhere. We put Actigrtaphy monitors on their wrists, and we measured sleep fragmentation during the night. And what we find, you can tell, is loneliness predicts the amount of sleep fragmentation, that is micro-awakenings, and it predicts above and beyond depression, anxiety, stress, and standard demographic factors. All right? Again, the detoxification being diminished over a lifetime can lead to a lot of toxicity in the system. For instance, these are the kinds of consequences that are associated with sleep debt. Metabolic syndrome, neural alterations, hormonal alterations, particularly in cortisol, all mimicking the effects of aging. All right? And so we think this to be one of the important mechanisms by which felt isolation is leading to early morbidity and mortality. But it's not the only mechanism. The largest cause of death continues to be cardiovascular disease, and so we've studied the autonomic nervous system. And in particular, we've looked at vascular activation. This is the results from a study in our undergraduates in response to a social stressor, a speaking task. And what you see is that peripheral resistance, vascular resistance, is higher in the lonely than the non-lonely participants. Now, blood pressure is what's called a regulated physiological endpoint. That means it is so important to maintain within a narrow range. Other things are going to get sacrificed to keep it within that narrow range. There's a handful of variables that are regulated. Blood pressure is one, body temperature is one, blood sugar is one, amount of oxygen going to the brain is one. If those things go off, you're in big trouble, right? So they're protected. Those homeostatic mechanisms decay with age. They wear out. They don't work as well. That's why you don't see hypertensive 20-year-olds. You do see hypertensive 50-year-olds. Vascular resistance is the best predictor of who develops essential hypertension. So do we get that? Well, first of all, I wanted to know, are we getting this effect only because they're in the hospital? So we put an ambulatory blood pressure and impedance cardiograph on these kids. We made these very small, and they walked around during a normal day into classes, and they were beat 9 times randomly. They sat down, filled out a diary, and what we get is the lonelier they are, the higher their vascular resistance. Now, cardiac output goes up to compensate, or goes down in our lonely, up in the non-lonely, to compensate for that. But this is the predictor of essential hypertension, so we then went to our older adult sample, and what we find is the lonelier they are, the higher their blood pressure in year 1. These are the variables we're controlling, all right? So you then ask, okay, well that's compelling in the sense of it's a correlation, but what's the evidence that it might be causal? So I turn to our structural equation, longitudinal cross-lag panel analyses, and here's the results of that. The lonelier you are, the bigger the increases in blood pressure. It's about 0.6 millimeters mercury per year per standard deviation of loneliness. So by year 4, we're seeing significantly larger elevations, cumulative elevations in blood pressure based on earlier levels of loneliness. All right? Now, let's go to neuroendocrine because I want to kind of get through some of these and leave time for questions. We looked at both sympathetic adrenal medullary, the catecholamine, your adrenaline system, and also the hypothalamic pituitary adrenal cortical system. This is the cortisol, ACTH system. We find in our young adults higher morning levels of ACTH, adrenocorticotropic hormone coming from the hypothalamus, activating the adrenal cortex to produce more cortisol. When we look at our older adults, in fact, we get higher morning rises in cortisol in our lonely older adults and flatter diurnal cycles. What that means is their HPA system is jacked up and it remains high. It's not getting turned off toward the evening. All right? Now, as in all the studies, we look both for not only that association, but specificity. Then we look for longitudinal, try to rule out. And in this particular study, we found 2 variables to be related to this morning rise. It was feeling lonely and feeling fatigued. So then we turn this around, we do the cross-lag. And what you see is the day before feeling of isolation predicted the morning rise in cortisol above and beyond these other psychosocial variables. How big the rise was didn't predict how lonely you felt that night. Daytime before fatigue didn't predict the morning rise. That morning rise predicted feeling less fatigued. Well, one of the things cortisol does is to break down fats and sugars to give you energy. So this is actually a known relationship in physiology. That's one that would be expected. If I give you dexamethasone, a cortisol surrogate, I'm also going to increase, kind of your energy levels. Okay, well, what about gene transcripts? Are we actually seeing the social regulation of gene expression? All right? And we published this first study in 2008 using literally a handful of subjects. I did the first gene transcript study on this topic in the late 90s, and we could do one. We could do one gene transcript. By 2008, we could do 3,000, but the assays, each one cost more than an fMRI scan. They're very expensive. At least they were in 2008. So we used 14 subjects, matched on everything except for their level of lonely. They were high and low lonely, but otherwise matched. And what we found was about 300 genes that were differentially activated. Most of them were turned off in our lonely rather than turned on. Now, those are a little bit like in imaging, looking at voxels. What you then do is you use those transcripts and you introduce it to a bioinformatics tool. We used TELUS. And what the bioinformatics tool, what you're doing with that is saying what are the coherent functions that are being served by these transcripts? All right? And when we did that, we found a handful of results. We found that the glucocorticoid response element transcripts, these essentially are the proteins that are carrying the signal from the cortisol receptor on the leukocyte on your immune cells and transporting that signal to the nucleus of the cell. That's downregulated. Now, besides breaking down fats and sugars, cortisol also is anti-inflammatory. And it's anti-inflammatory by its influence from this cortical signaling to the nucleus and then what that nucleus does in terms of gene expression. If I've decreased that breaking power, I'm going to increase the production of pro-inflammatory transcripts. Those are called NF-kappa B transcripts that then result in pro-inflammatory cytokines like interleukin-6 and IL-1 beta and TNF alpha. All right? And so we get that kind of mix of decreased cortisol regulation, increased glucocorticoid insensitivity, and increased organismal inflammatory biology. We also get a couple other effects. Get an upregulated response to oxidative stress in our lonely individuals. Remember, that was the mechanism that killed the fruit flies. And we get downregulated type 1 interferon innate antiviral responses. All right? Now, that may sound like a far reach in terms of what's common, but there's a coherent story to this. We more recently replicated this with the remaining part of the sample that would consent to genetic studies. And we looked at whether it was related to depression or social support or objective social circumstances. It was only felt isolation that was related to these gene transcripts. We then looked at which cells in the immune system were responsible for this effect. This is our discovery sample. This is our confirmation sample. And what you see is it's the oldest cells in the immune system and the newest cells. These cells, monocytes and dendritic cells, evolved early because they protected against bacterial invaders. All right? Bacteria doesn't need you at all. It's an organism that can live on its own. It's sitting on that claw or that tooth or that fence or that tree that you scrape or cut yourself on, and then it gets entry into your body. Right? If you're alone, or if you're in a hostile context, the pathogen to which your immune system has to worry are bacteria, right? Because it enters the body through cuts and scrapes, and it doesn't need others for transmission. If you're in a socially affine circumstance, now the pathogen to which I better be careful about are viruses, because viruses get transmitted through body fluids. I sneeze, I shake your hand, I sneeze, I shake your hand, you scratch your eyes. That doesn't happen if we're not close or if we're fighting, all right? So your immune system actually changes depending on the social context. If you feel it's hostile and you're isolated, you get a shift in gene expression to protect against bacterial invasion, and the opposite if you're in a socially affine circumstance. So that seems to be the story underlying the gene expression changes we've seen. I wanted to show you that we've only touched some of the effects. There's a much broader set of findings we and others have found, and all of the effects I've mentioned are being replicated elsewhere, often in cross-sectional rather than longitudinal studies. But these associations that I've shown you seem to be holding up in other countries and in other laboratories. This is to show you the effect size of what we're talking about. Loneliness predicts morbidity and mortality. How much so? Here's the effect size for obesity. Here's for loneliness. All right? So it's about 2 to 3 times larger than the effect size for obesity. So if we think obesity is an epidemic, that's a major health cost that's going to be a problem for our country, we have to think about loneliness. Now you might say, but wait a minute. Loneliness is just the problem of a few, and obesity is the problem of many. Not really. Let me show you America. And this is the percent of single household residences in 1940. This is it in 1970. All I have to tell you is the darker the blue, the higher the number. This is in 2000. I looked at 2010 census data. It's increased yet again. Right? Here, Utah is blue. Not anymore. Not anymore. It's a darker blue now. All right? Now, objective isolation is not the same as perceived isolation. So we did a study with AARP, a nationally representative sample, because they wanted to know about loneliness across the nation. And what we found was 40% of older Americans report being lonely, frequently and highly. What I was using as a cutoff for high and lonely in the top quintile just 20 years ago in our undergraduate study. All right? I thought that was high. And I looked at the data to find some artifact that could explain it, and I couldn't. And more recently, a study was published this summer showing 43% of older Americans. They had an older population than we had. We had 45 and older. They had, I think it was 80 and older. 43% feel isolated or lonely most of the time. So these numbers seem to suggest loneliness, in fact, has increased due to these other differences in how we're living, and are in fact high and prevalent, as we're seeing with obesity. So it's a big effect size, and it's prevalent. And with that, I want to leave time for questions. This is simply to show you that the question of looking at how we are as an individual takes, in fact, the work of a village, an international village. And I have this group to thank for the research that I spoke about. And I want to thank you for your time and attention. Happy to answer any questions you might have. Yes?
Wonderful stuff. I saw on one of the recent slides, briefly mentioned some of the functioning.
Yeah. Sure. What we found around late 90s was that loneliness was compromising executive functioning. In particular, what it does is it increases impulsive behavior. It's not, executive functioning is a heterogeneous lot, right? So specifically, it's impulsive behavior that's increased. In our CHASRS study, we find loneliness predicting who's exercising and who stops from exercising, right? We looked at whether it was social control or it was executive functioning, and we found executive functioning, not social control. We found it both cross-sectionally and longitudinally. We also find lonely individuals greater percentage of fats and sugars in their caloric intake, right? Comfort foods, right? When you look at non-human animal work, they find, isolate the animal, it increases, quote, prepotent responses. What's a prepotent response? It's an impulse, what we would call an impulsive behavior. It's the highest on the hierarchy, right?
I think that the causal direction there is that the lonely people develop more impulsive.
That's what our longitudinal analysis suggests, and that's what the animal studies suggest. Roy Baumeister has also done work where he's made people feel the future alone, and he finds that increases impulsive behavior as well. I mean, he talks about it in terms of executive functioning, but if you look at the measures, they're going for the yummy food rather than the nutritious food. I call that prepotent responses rather than executive functioning writ large. So that's what it seems to suggest. Nelson.
I have a question about what it is about loneliness that might have the effect. So it could be the absence of social interaction, or it could be, for example, an attribution that other people don't value you, and those would be very separate in different individuals. Have you thought or looked at that?
Yeah. So each of these components that I mentioned weight that differentially. And so we've wondered if there are differential consequences of these different phenotypic expressions of loneliness. So far, we've not really identified anything that I would want to bank on as distinct. I mean, the very first one seems to be more related to cardiovascular, vascular resistance than the other two components. But it's not as general or profound as I might have expected, as you might have expected with that question. It seems to be what's missing is, and again, I want to put this in terms of what the brain is surveilling for. We, as a social species, need others with whom we can work and overcome life's challenges. Moving a table by ourselves versus with someone, it's more than additive, right? There's just this game-changing. And as a social species, that seems to be critical. When you don't have those conspecifics around you can trust and work with, the brain goes on alert for it's in danger. Self-preservation kicks in. It looks for connections, but it looks for connections in a kind of dubious, potentially paranoid fashion, if you will. And I think that may be what's actually critical here. Now, there are attributional effects. When we look cross-culturally, surprisingly, collectivist cultures, loneliness is higher than an independent. And you might think that's surprising until I ask you, when do you think loneliness is highest across the year in America? It's at the holidays, when culturally you're supposed to be enjoying friends and family. You may be with that friends and family. If you're sitting there feeling hassled and bedraggled and knowing that you're supposed to be having fun but you feel isolated, that's a very, very alienating experience, right? If I've got work to substitute, that loneliness isn't very salient. So that's an attributional process. I should be enjoying myself and I'm sitting here miserable. I have done hundreds of interviews now on phone, and I talk about the science. The one exception was a phone interview done during the Christmas holidays. And although I was trying to talk about the science, none of the callers were interested in that. They all were telling me about their personal angst during that period. So the attributional processes contribute additionally. It's just before we thought it was all that. And going across these animals and the studies I've shown you, I think it's much more fundamental, and then you get elaborations by the attributional processes. But there's actually qualitatively different in some of the effects of them. Steve.
Are sociopaths people who don't want to be around other people, who don't like other people?
Yeah.
Are they not vulnerable to some of these?
That is right. They are much less vulnerable. In our rhesus monkey model, there are three groups of monkeys. Those socially gregarious monkeys, the complete isolates, and the monkeys that perform what are called walk-bys. They walk within an arm's reach of other adult monkeys, and then they walk away, and they do it repeatedly. They play with juveniles. They walk toward adults, and arm's reach with the rhesus monkey is an important measure because that puts them in danger. Another monkey can grab them and bite them and harm them. So they actually put themselves in danger and yet don't consummate the social interaction. They seem to be, at least this is work with John Capitanio, who's at the California National Primate Center. He considered them the loneliest. When we use that behavioral category and look at our human adults, they're the loneliest. The gregarious are the least lonely. The isolates are in the middle. When we look at hormones and gene transcripts, it looks like, and we're still in the process of this analysis, it looks like the animals are aligning with our humans along that same continuum. So being an isolate itself doesn't put you at risk. It's whether you choose to be an isolate. When people grow older and they stop being able to go, when they're alone but not because they're choosing to be alone, that's when we find objective social circumstances predicting loneliness. It's only then that we find whether they're physically alone being really related to whether they feel lonely. So it has to do with control or choice. Yes, and then you. Yeah. So in our hypnosis study, there's an important point I should make because it answers your question. We could take those in the middle and make them like top quintile, right? We can take them and make them like the bottom. We could push them about a third of the distribution. Those in the bottom in loneliness, we could move to the middle. Those who are very lonely, we can move down to about the middle. So it's not like you can take someone and transform them to the other side of the distribution. There seems to be some reaction range, if you will. But it's much larger, and the effects are much more profound than I expected when we were, and I could ever have hoped when I first looked at our cross-sectional. What I thought were big individual differences, we see these dramatic effects within people when they have these profound changes in social contexts. So that's a quick answer to your question. You, I'm sorry.
Yeah, I was just curious about what you thought, how social media might contribute to loneliness.
Yeah.
Just based on age and...
So this is basically your question? Yeah, well, we're living in a very different social environment. And so it's a very reasonable question. How has social media changed this? I want to point out that the social media is a tool, right? And so as a tool, its effects should depend on how you're using that tool. So if you look at the literature, it's really kind of muddy. And so we had a simple hypothesis. If you use the social media to enhance face-to-face interactions, it'll be associated with lower levels of loneliness. If you use it as a substitute, it's a little bit like being hungry and eating celery. It feels good for the moment, but ultimately it's not nutritious, so it does more damage than good. All right? So what we did was we looked at the percentage of the interactions with confidant were face-to-face or through the social media. And what we find is the greater your interactions are face-to-face. And these are undergraduates. They're all using Facebook, right? The greater the percentage of face-to-face, the less lonely. I mentioned my son just posted on his Facebook, is there anybody who wants to go out and drag tonight? Okay, that's not what your father wants to see you posting. On the other hand, you can see he's using Facebook to increase this contact in ways I don't approve, but all right? If instead, though, it could have been worse is the answer. If he's using it instead as a substitute, so this is where the greatest percentage of the interactions are online, you see all of those are associated with higher levels of loneliness. Unfortunately, lonely individuals tend to use it as a substitute, and they do a second thing that makes us even worse. They present non-authentic selves. So if their authentic self can't get affirmation in face-to-face contact, they're worried it won't even be affirmed in internet contact, so they're presenting a false self. So you can imagine, you're finally affirmed, but it's not actually you. So they're the ones at greatest risk for loneliness. I'm sorry.
To what extent is loneliness acquired versus inherited?
Great question. Yeah. The answer to your question is loneliness is 50% heritable in adults. In children, it varies. In adults, there's no common environmental variance. It's all non-common. In children, there's common environmental variance. That's why the heritability goes down a little bit. All right? That 50% isn't feeling lonely. What it is what is your pain sensitivity to social disconnection? We need that variability in our population. You need people who are so pained by disconnection that they will say and defend the village against challengers, right? You also want some, though, who aren't so pained by disconnection that they're not willing to go over that hill and explore. If they feel no disconnection, they won't come back. So you want some disconnection feeling, right? So that they explore, but they're going to come back and share. Lewis and Clark, if they had just gone out and never come back, wouldn't have been very useful to the village, right? But as a distribution, you also expect some at the extreme who feel no pain at all. And I think those are psychopaths. I don't know for a fact, but that's what I actually fear characterizes a psychopath, is they feel no connection at all. And they, and as a result, so you see, one of the things I'm suggesting is loneliness contributes to our humanity. Because if I don't feel that connection with anyone, I'm capable of terribly inhumane things. It is that pain that promotes this connection with other individuals, and that connection is what promotes our humane behavior toward one another. So in an odd kind of way, loneliness makes people not very pleasant to be around. But I'm also really grateful for this social pain system, if you will. You, then you. Go ahead, waiting.
So I was wondering, earlier you mentioned that people who are lonely have this reduction in social skills because
Yeah.
you're on the periphery
Yeah.
and you're thinking about
Yourself, yeah.
You're not really worried about other people. But
Yeah.
thinking about other people is how you get back into the fold, though, back
Yep.
into the
Yep.
center.
Yep.
So I was wondering about.
So here's the, you got to put in an evolutionary context how we used to live and how we live today. One of the things, if you've ever spoken to an individual at a cocktail party, an individual who's very lonely, you probably felt like you were being eaten alive. They hunger for human contact, and so it's like, whoa, I need to get out of this circumstance, right? And so part of the clue, when I have 5 seconds, you're in these phone interviews, they say, well, so how would you tell somebody if they felt lonely? And I say, do volunteer service, because you've put your finger on it. Being connected isn't about people doing things for you, it's about having synergies between people, right? And so getting outside of what they can do for you and looking at what you can do together, taking that person's point of view, not being generous at your expense, but being generous with the other person. So the reason I say go volunteer, instead of eating other people, feed them, and you'll see the positive reciprocal actions from them, and that will perhaps lower the threat and self-preservation and self-focus that results. But it is not we've done a meta-analysis of all the studies that have been designed to lower loneliness. This is not an easy enterprise, but I will say providing social support, providing social skills training, providing social engagement and opportunities have very small effects. What seems to have the biggest effects are actually addressing some of the issues we've been talking about, changing how it is that they think about other people, changing not the focus on themselves, but changing it to be a focus on them and other people and on the synergies that can be achieved. That seems to be the best way, but the devil is in the details of those interventions. You had a question.
Yeah, you make a very convincing case that loneliness is this epidemic, kind of like obesity. But when we talk about obesity as an epidemic, we tend to talk about like prevention and intervention efforts.
Yeah.
So what would a prevention or intervention effort for loneliness look like,
Yeah.
particularly with adults? And how could you sell a skeptical funding climate and a skeptical public on some kind of intervention for loneliness?
So one of the things I didn't show you is loneliness is contagious. We've done a study in Framingham, Massachusetts using the Framingham data set, and we found that loneliness actually gets transmitted across people. Even so, if I'm a friend with you, but I don't know the person behind you, but you're their friend, it gets transmitted even to people I don't know. And it occurs because of the behavior that it inclines. Remember that cycle I showed you? If I act nasty toward you and we're friends, we're less likely to be friends in 3 years, right? But my nasty behavior toward you increases the likelihood you're going to mistreat some other friend. And so it actually gets transmitted across people in that fashion over years, all right? So what kind of intervention? Well, how about selling the U.S. Army and trying to do something about the isolated feelings of their soldiers? Isolation is part of a soldier's life, right? And so we've been working with the Army. We were at Fort Sill 36 hours ago, talking to a general there about this. And we've been training soldiers and platoons to build social resilience, with the whole idea being that when they go into war, they're better able to support one another and come back in better shape than they currently are coming back. And that intervention program tackles, for instance, that what it means as an infant or a young child is to be dependent on others. What it means to be an adult is not to be independent, which is what the typical answer, like, what does it mean to be an adult? Well, I'm dependent now, I'm going to grow up and I'll be independent. And God knows the Army has kind of built that culture. But that's actually not the right answer. To be an adult in mammals is to be the animal on whom others can depend. And that's a profoundly different view of what your role is as an adult within a family or within an Army platoon. And so we do a lot of training of those kinds of things. We do mind reading, teach them how to read voice and eyes and facial expressions, but then point out that you always have to test, and we teach them how to verify. So it's a little bit like being a little scientist about your social interactions behavior, teach them about behavioral confirmation processes, blah, blah, blah. So basically trying to train them, we give them skills, but it's not so much those skills as it is to get outside of your own perspective and take the perspective and work with others. And then we also do training of the platoon. Given loneliness gets translated into other people, the platoon itself has an obligation to every soldier within it. So that you see you're training at multiple levels. And that seems to have worked. That seems to be working. The effect size for that is about what you see in the effective intervention studies that we've reviewed with individuals. Okay, one more question if there is. Yes?
Every neighborhood basically has a cat lady. Has a cat lady. Are these symptoms alleviated by cross-species?
Yeah, so we actually, the short answer is the poor cat lady. We've looked at what's the benefit of pets, and I never knew people had such weird pets as we found in Chicago. But dogs are associated with lower loneliness. Cats, no different. Fish, no different. Reptiles, no different. Hamsters, no different. Birds, no different. It's really dogs. Now, that's kind of interesting because if you know the coevolution of dogs and humans, some biologists will tell you dogs have made us dumber and we've made them smarter. They're the only animal that does social referencing. They follow eye gaze. Even chimps don't follow your eye gaze. So they're keenly attuned to our social signals. I don't know if that's critical. I don't know if it's because I go out and I walk my dog and I meet other people. I don't walk my cat, right? So I don't know which of those is the reason. Either one may be the reason, but dogs are associated, actually seem to have some benefits. So you know the old politician story, if you go to DC and you want a friend, get a dog? This actually seems to be true. Okay.